Hello to everyone, my name is Eglantina Idrizaj, and I would like to talk about the effects on the gastric motility of two adipokines, adiponectin and resistin, and if there is a possible correlation of these their effects with food intake. Well, first of all, we should say that the regulation of food intake is quite a complex mechanism that involves the interplay between central and peripheral structures. In fact, hypothalamus, which is a key region of the regulation of food intake, receives and integrates signals not only with other central areas, but also with peripheral organs such as gastrointestinal tract or adipose tissue. In fact, hormonal signals originating from the gastrointestinal tract or adipose tissue could reach hypothalamic nuclei and influence food intake. In this view, gut hormones such as cholecystokinin or GLP-1, as well as hormones deriving from the white adipose tissue, which are called together as adipokines, and we can mention here the well-known leptin, are reported to induce anorexigenic effects by acting centrally. But in the last years, the scientific focus has been directed in the study of other two adipokines, adiponectin and resistin, which have been reported to reside within the same vesicles in the mouse adipocytes and to be co-secreted in response to the same triggering signals. Well, among their different actions, adiponectin and resistin are shown to have a role in the regulation of food intake by inducing central anorexigenic effects. But notably, many hormones which act at the central level are also able to affect gastric motility, and in this view, we previously reported that both adiponectin and resistin were able to exert inhibitory actions on the gastric motility in strips from the mouse gastric fundus. Well, such inhibitory actions could, of course, lead to gastric relaxation, which may lead in turn to gastric wall distension. Gastric wall distension triggers stretch and tension, thus stimulating mechanosensitive receptors and activating in turn vagal afferent nerves to transmit their information to several brain areas, sending so peripheral satiety signals. So we hypothesize that the effects of adiponectin and resistin on the motor activity of the gastric fundus could represent actually peripheral satiety signals which may concur to their central anorexigenic effects. But furthermore, we previously observed that such inhibitory effects of both resistin and adiponectin at the gastric fundus level appear to occur at least in part through a nitric oxide-dependent mechanism. Of course, nitric oxide is considered as the main inhibitory neurotransmitter released by the non-adrenergic, non-cholinergic fibers supplying the gastric smooth muscle and responsible for relaxation in both humans and rodents. In particular, neuronal nitric oxide isoforms appears as the main isoform able to produce nitric oxide as a neurotransmitter, which play the most important role in the control of gastric motility. So, in this basis, the aim of the present work was to investigate whether the hormone actions, so resistin and adiponectin at the level of the gastric fundus, could mutually enhance each other, and if neuronal nitric oxide isoform was involved in their effects. So, to this purpose, the combined actions of the two adipokines on either the neurally induced relaxant responses or on the neuronal nitric oxide synthase isoform expression was evaluated in preparations from the mouse gastric fundus. So to this aim, we use mechanical technique to record the effects of the hormones on the neurally induced relaxant responses, and to do this, longitudinal isolated preparations were mounted in organ bath containing Krebs henselite solution. and connected to force displacement transducer, which are coupled to a polygraph for the continuous recording of the isometric tension. Then neural-induced responses were elicited by applicating electrical field stimulation at two stimulation frequencies 4 and 8 Hz, which was applied via two platinum wiring through which the preparation was threaded. Then, to put in evidence the inhibitory component, so to activate the non-adrenergic, non-cholinergic fibers, we first of all added to the bath medium carbachol to exclude the excitatory influence, because during electrical field stimulation, both excitatory fibers, which are mainly cholinergic, and inhibitory nerve fibers, which are mainly nitrergic, are simultaneously stimulated. So we applied electrical field stimulation in carbachol precontracted strips, and we observed relaxant responses which persisted during all the stimulation frequency and which increased by increasing the stimulation frequency. Then we added to the bath medium tetrodotoxin, which is a nerve blocker, or L-nitroarginine, which is a nitric oxide synthase inhibitor. And what we observed was that in the presence of both tetrodotoxin or LNNA, the inhibitory responses were abolished. So indicating the nervous and nitrergic nature of the electrical field stimulation induced inhibitory responses. Then we tested the effects of adiponectin on the neurally induced relaxant responses, and what we observed was a statistically significant increase of the mean amplitude of the relaxant responses at both stimulation frequencies employed at 4 and 8 Hz. So such effects were already detectable in 15-20 minutes after the inclusion of adiponectin to the bath medium. And then after 30 minutes, we added to the bath medium resistin, and what we observed was that actually the inclusion of resistin did not cause any further increase in the amplitude of the relaxant responses. So in a second set of experiments, the effects of resistin were tested, and we again observed a statistically significant increase, which actually appeared more pronounced, but it was not statistically different compared to the response obtained by adiponectin alone. And after 30 minutes from resistin, we added to the bath medium adiponectin, and we observed that, however, the inclusion of adiponectin to the bath medium after resistin did not cause any further significant increase in the amplitude of the electrical field stimulation induced relaxant responses. So, to confirm the above observations, we also tested the effects of the simultaneous addition of both adipokines, and in these conditions, the mean amplitude of the electrical field stimulation induced relaxant responses was significantly increased, but this increase was not statistically different from that observed when the hormones were individually added to the bath medium. So, analyzing this data, the observation that in the presence of adiponectin, the subsequent inclusion of resistin in the bath medium and vice versa, as well as the concomitant presence of the two hormones to the bath medium did not cause any additional increase in compared to the effects of each hormone alone, indicates that the two adipokines exerts their effects at the level of the gastric fundus on the nitrergic neuron transmission by engaging the same mechanism. Actually, it was quite a surprising result to us, because given the similar actions exerted by the two adipokines at the gastric fundus level, and the ability of the mouse adipose tissue to co-release the two adipokines, it was plausible to expect that the effects of adiponectin and resistin might actually aim to strengthen each other. But however, also Western blotting and immunofluorescence experiments, which were performed in collaboration with the histological section of the University of Florence, and also this analysis confirmed the above results. In fact, Western blot experiments put in evidence that both adiponectin or resistin induced a significant increase in the neuronal nitric oxide synthase expression, and that no additional increase was observed in their concomitant presence. Such results was also confirmed by immunofluorescence analysis too, which revealed an increase in the number of the neuronal nitric oxide positive neurons in specimens exposed to both adiponectin or resistin alone or to the concomitant presence of both hormones, and no differences among groups were observed in these conditions. So, in conclusion, the functional and immunohistochemical experiments put in evidence that the effects of adiponectin and resistin do not reinforce each other, but that both hormones engaged the same pathway to exert the same actions at the gastric fundus. And in particular, they both determine a neuromodulatory action on the ninergic neurotransmission by inducing an increase of the neuronal nitric oxide synthase isoform expression, which appears as a shared target for the two hormones. And from a physiological point of view, the actions of the hormones at the gastric fundus level could actually represent a dual redundant mechanism useful to guarantee an adequate amount of nitric oxide production and at the same time to avoid unaltered, excessive or defective production of this neurotransmitter, which may lead to pathological conditions. So further underlying the importance also of this neurotransmitter to maintain gastric relaxation. Indeed, the inhibitory actions of the hormones at the gastric level may lead to gastric relaxation, leading in turn to gastric wall distension that, through the activation of afferent vagal fibers and the interposition of nucleus obturatus solitarius, could reach hypothalamus, so generating peripheral satiety signals. Thus, a better understanding of these mechanisms may represent the basis for the treatment of some pathological conditions, such as gastric dysmotility, or even in some alterations of food intake control, of course. Moreover, I just want to introduce some of our forthcoming results which support the role of adiponectin to generate peripheral satiety signals, which, in addition to the stomach, may also originate from the small intestine. And in this view, our data still to be published showed that adiponectin was able to induce an inhibitory effects also in the ileal motor activity, an effect that from a physiological point of view may be addressed to slow down intestinal transit time, so prolonging the permanence of nutrients in the more proximal regions of intestine, thus inducing an increased release of the anorexigenic hormones by the enteroendocrine cells, as also occurs in the physiological ileal brake reflex, and therefore the inhibitory effects exerted by adiponectin at the ileal level could represent also an additional peripheral satiety signals. So I would like to conclude that even if caution is needed on transferring data obtained from animals to humans, the above observation could provide a stimulating starting point for a better understanding of the mechanisms underlying the role of adipokines in the gastrointestinal motor responses related to some pathological conditions. I would like to thank my mentor, Professor Bakari, the organizing committee for inviting me to share with you this online presentation, and of course, thanks a lot to all of you for your attention.